All right, folks, let's get this party started. Hello, everybody. Happy Tuesday. That's right. I don't know how many people say that. Happy Tuesday. Come on. And it's better than being outside right now. So, welcome to the Grit City Think and Drink. I'm the host, Jim Gall. I'm a professor across the street at UW Tacoma. Welcome. How many people are new to the Think and Drink? Hey, awesome. Glad to see you guys in. Just to let you know, so this happens the second Tuesday of every month, except during the summer when it's about 95 degrees back here, and those fans don't do a damn thing. So we take a break during the summer, but we have talks set up through June and then starting again in October. So we hope you'll come next month. Ellen Moore is going to be talking about her research in Hawaii, where they use water as a way to kind of think about how they manage things on Maui. So it's a really cool talk. Hope you'll come for that. And then in June, Cheryl Greengrove, who does oceanography, will be here to give a talk about her work over the years at UW Tacoma. So I hope you'll come and check us out. If you want to see what's coming up, you can scan the QR code and put your email in there. All I send out is announcements about the talks, nothing else. So if you want to do that, you can check that out. Or you can go to the Grit City. If you Google UW Grit City Think and Drink, you'll come up with that, and you can see what's coming down the pipe. And you can go back and look at recorded talks that you may have missed that you might be interested in if Jim didn't screw up the AV equipment and it doesn't look anything like what it was supposed to. So that's the way it sometimes goes. So the way it works is I will introduce our wonderful speaker. He will give you a talk to knock your socks off. And then we'll give a short break to the speaker. We'll give away, if you haven't already, we'll give you a raffle ticket and you can win swag. So we're out of t-shirts at this point, but we're giving away bandanas with the logo on it. So that's next. And then after that, we'll do a stump the speaker, ask questions afterwards. Sound good? All right. Well, without further ado, I'll introduce our speaker. Morgan Heinz has a background as an evolutionary biologist mapping reptile diversity and is now an assistant teaching professor at UW Tacoma. I don't actually know how long I've known Morgan. It's been quite a while now. Yeah, it's been a while. Coursework Morgan teaches for science majors include coursework Morgan. Punctuation. Damn it. Coursework Morgan teaches for science majors includes the intro bio series, biogeography, vertebrate diversity, and anatomy, and the occasional study abroad framework course. He also frequently teaches mixed majors, non-majors courses such as issues in conservation biology, genetics and society, and the nature of human diversity. He is the jack of all trades. Morgan was trained in educational professional development with HHMI, Interactive Ambassador Academy, and facilitates collaboration with the international community of high school and college expert educators to share and implement evidence-based teaching strategies. And he is an advocate for the Bio Blitz, which he will talk about tonight. So without further ado, Morgan.
What Jim might not have mentioned is that hopefully there's also going to be an interactive portion for this. So now I just have to move my slide deck. Oh, how do you move it over, Jim? How do you move? Excellent. So here I have a couple of photos of myself. If you'd asked me to describe myself, it would have been a lot of those words that Jim just said. As an evolutionary biologist, my field of study for a long time was herpetology, reptiles and amphibians. So you can tell these are all photos of me when I was younger because I still had all of my hair, but I've got a bunch of different lizards that I was looking at. What I'd like to talk with you today about, though, is how some of my focus has shifted from the critters that I can catch in my hands that are lizards to a wide variety of other organisms, and how that was facilitated in part by my moving here to the Pacific Northwest. Right. So when we're out in nature, you might think of, all right, the person who's going and documenting nature and finding all of the cool things might need a whole bunch of supplies to be able to find the critter they're looking at, to get it in their hands, to examine it, and to identify it. So often we think of things like binoculars and magnifying glasses, microscopes, and then, of course, field guides that have all of this expertise in them. Is it more like this photo, or is it more like that photo? Peterson's field guides famously started putting in those images of the illustrations like little arrows. Look for the neck to be either white or gray. And that was like a revolution in identifying organisms. So there's been a whole series of sort of field guide idea and design for trying to communicate some sometimes pretty subtle differences among species so that we can find them ourselves, learn to enjoy them by putting a name on them. And sometimes we also think about, okay, maybe there's some specialized equipment that you might have. For example, what I've already dropped, here. I've got that white sheet hanging, and so sometimes I will put up a sheet like that. And then I'll come back a couple hours later, and on the UV sheet, there'll be a bunch of moths. And it's like, they're doing all of the work for me. It's great. But I had to have the foresight to go think and set all of that up. Or I might just find a white light on the side of like one of the bathrooms or one of the metro parks around here and know these places to look for organisms. Or there might be some more technique or approaches involved. So for example, often people who are looking for insects will use what is called a beat sheet. And I also use that fabric for that. So you sort of hold it out. Some people use an upside down umbrella. And then you just whack the heck out of a tree and all the insects fall out of the tree into the little dish and you get to see the insects. And then when you're done, you can sort of shake out your umbrella and let them go. You might think of, okay, well, if we're doing experiments with organisms that we need to see up close that are kind of harder to hold in the hand, we might have, for example, some equipment that we need to set up here, some saltwater setups that I did as an undergraduate student at a research station in Belize. And sometimes this does take a little bit of ingenuity, but it's generally a fun and exciting and engaging kind of interactive. And here's a little bit more of me in Belize. But what I hope to impress upon you is that all of these different techniques and all of this different equipment has been completely overwritten in my mind by one tool that has changed the way that I interact as a naturalist, as an evolutionary biologist, as a biogeographer. And I would like to invite you to also partake of it. Because I would say the number 0 tool is curiosity, right? Caring about the world around us. You have already shown some curiosity by showing up here, unless you just happen to randomly walk in. Seats are nice. And then the next tool that we have that has revolutionized even more than those field guides with the little arrows in them is absolutely our cell phones, which have cameras and GPS in them. So today, as Jim hinted, I'm going to enthuse about a particular application called iNaturalist that has steadily taken over my life in what has been, for me, a very positive way. And my hope is that you will get a chance, if you have not, to try iNaturalist, to see how it works with some of the specimens that I brought for you to hear today, okay? So iNaturalist works on iPhones and Android phones. It is free. It is, as far as I know, it will always be free. And like Jim was saying, I don't get annoying emails from them kind of thing either. So if you do not have iNaturalist, I invite you to stop looking at me or the screen and do the thing we don't do in class. If you would like to try installing it, you may. So I didn't grow up on the West Coast. I grew up on the East Coast. I studied zoology at North Carolina State University. I came down to San Diego to pursue some research there, and I eventually came up to Seattle. So when I moved to the Pacific Northwest, a lot of organisms were very new to me. Right? I was coming from North Carolina where there are tons of kinds of salamanders and snakes, and the Reptile Amphibian University here is very cool, but it's many fewer species. So I started going on walks with my dog and hikes and realizing I don't know anything about this ecosystem. So as I slowly began to learn, iNaturalist was one of the tools that helped me learn to recognize the biology around me. So for example, does anybody know what this plant is? Yes, what is it? This is salal, or Gaultheria chalone, if you want to use the scientific name for it. Salal is an understory plant that's very common around here, but I took this photo in January. And the reason I took this photo is I got used to seeing salal everywhere, but I noticed that it was blooming. And I was like, I've only been in the Pacific Northwest for so many years. Is this normal for this plant to be blooming here or not? iNaturalist is going to help us answer that question, but not yet because you're still installing it. Okay, so here's how iNaturalist works. Once a person has the app on their phone, or you can use it on desktop with like a big DSLR camera, all of that too. You've got an image of an organism, or hopefully multiple images of the organism. You can also do a sound recording, such as of a bird call. You upload that to the app, and here's our example specimen. You can take a couple of photos, again, from a couple of angles is more helpful. And then the app is going to analyze those images and provide a best guess as to what the organism is. So the best guess here is it's a placental mammal, which is accurate, but not like You'll also see that canine, like a domestic dog, is the next best guess. So this is the sort of the core loop in this software. You see something, you document it. It can tell you its best guess at what it is. But then you upload, if you choose to do so, your observation to a database that now has over 300 million global observations. And that includes the GPS coordinates and the time you saw it. Right? And then comes the other part of iNaturalist. It was designed not just to be a database that helps you identify things, but it is a social network. And it's the only social network that I am on. And I suddenly understand why social networks are so addicting, because once I have uploaded my photo, a bunch of other naturalists in the area, shout out to Mimi there in the back, she's awesome, a naturalist, will come along and they'll be like, oh, hey, that's kind of unusual for that plant to be blooming right now. Did you put the wrong date? Or they'll help me identify something. So here's another example. Oh, and here is the data from around the world. This is representing those observations that are present. It is very, very much, and not 3,000 million, 300 million, that's a typo, records. Now, depending on the quality of the image, again, the computer vision may be more or less certain. So on the left, you'll see that that specimen is immediately identified to genus very confidently. In the middle, there's a good guess, but those leaves could be a lot of different kinds of monocots. On the right, the computer vision's not going to know what that is, okay? So when we upload the images, sometimes it'll be like, we're pretty sure this is a touch me not, right? That's probably impatience. The one in the middle, we're like, okay, is it the daylily or is it the insect that eats the leaves of the daylily? I'm not actually sure which of these two it is. And on the right, it's either a black oak or a spongy moth or a yellow-bellied sapsucker bird, right? Which is to say, the computer vision needs more information. Maybe if we get more photos from different angles, it'll have more information to make a guess, or just upload it to the network, and then other people will be able to use their expertise. Right? So here's a spider that I found in Lakewood. I was like, I don't know what kind of spider it is, but I know it's a spider. So I uploaded it as a spider. And then along comes this fellow, and he's like, oh, that's Coriogonal monoceros. I was like, thanks, dude. Okay, I didn't put that I agreed because I can't identify that spider. We just left that tag on there for now. But now I have a name for it. And if anybody's looking at the scientific name here, you might notice something interesting about this spider. Monoceros. This spider has like a rostral facial projection on it. And so I could respond to that person and be like, I didn't even see that, and I was this close to the spider. Right? So now I have a name for this organism, and I have discovered something very cool about it. I was really stoked for this. And I have since seen this spider 2 more times. All right. So I was like, who is this Arachnologus who is bestowing upon me such wisdom? And so I clicked on his profile, and it is perhaps what you would expect for someone whose name is Arachnologus. This is Rod Crodford. He is an amazing person. I have never met him in person, but he really, really, really likes spiders. He is the curator of arachnids at the Burke Museum at UW Seattle. And to my knowledge, he looks at every single spider observation that goes on iNaturalist in Washington State. So as soon as I started getting this decades worth of professional super expertise looking at all my spider photos, I suddenly started taking a lot more photos of spiders, because I could put a name on something with Rod's help. And again, I've never met this fella, but I'm really grateful to him. Here's another example. I've never met Zachary Dankowicz or his brother Evan Dankowicz, but they are both prolific identifiers of flies. There are lots of flies in the world, and they are making an expertise out of it. But unlike Rod, they're pretty new to this game. They started doing this with iNaturalist as high school students. So here we hear from Zachary. I'm a junior in high school in Maryland. In December 2020, during my freshman break, I was just so bored. I didn't want to play video games, had nothing better to do than that. So my brother Evan, who was at home because of the pandemic, offered me something to do. And in the afternoon on December 23rd, I made my first identification on iNaturalist. Just as a high school student, was like, I'm going to try and figure out what one of these is and start from there. And since then, Zachary and Evan have gone on to create an online guide to help everybody else identify flies. And it is a very wealthy source of information, completely accessible to everybody. It's called The Fly Guide. Evan is now a research assistant at Harvard University, and Zachary is pursuing entomology and environmental studies at University of Maryland. Okay. Here's some other cool critters. And these are ones I hope to give you a chance to see. Once I started slowing it down and looking at the small stuff, I realized I wanted a few more tools. So again, while your cell phone, it's probably sufficient, I went out and got like a camera that lets me zoom in on really small organisms. And I started seeing these insects that are called springtails. They are ubiquitous in the soil. They are everywhere. And I had been flipping logs looking for snakes for decades and never saw them because I wasn't looking for them. But when I started to realize they were there and I had the camera to look at them, I saw that they were really quite beautiful. And they can also do something that's really fun. They're called springtails because they have an extra appendage underneath their belly that's spring-loaded and allows them to jump hundreds of times their height. This is very much slowed down. They can rotate dozens of times per second. These are very easy to find in the Pacific Northwest during the right time of the year under basically any log. And in fact, the springtails can also do this jumping kind of maneuver in a bunch of other environments. So I often find them under logs, but what you might not have noticed is that this springtail is taking off from water, right? So they are small enough and light enough to jump from a water surface. So here we have a really tiny one at the top that's much smaller than 1 millimeter, and then Isotoma is about 1 millimeter or 2 long. They can be incredibly dense. You can have lots of them at the right time of year. So this is a bunch of those longer springtails on the water. They can be on the marine intertidal. They can be freshwater. They can be under logs. And I hope that your life is richer for having known these springtails, right? Mine certainly is. Now I'm so excited to see them, and I'm glad that they are so many places to see. All right. So here is Alex Tinker-Salvas, who is another high school student. He's in Germany, and he's all about springtails. He photographs them a lot, and this is one of the records that he's put on iNaturalist. But the other cool thing about this photo is that it had won him the 2018 Young Wildlife Photographer of the Year award. So he's got a ton of expertise in taking photos of really, really small things, and he uses iNaturalist to identify those things. So I have learned from Alex a lot about macro or close-up photography, even though I have never met Alex because he's busy running some of these iNaturalist events over in Germany. So for example, he has a YouTube channel that I discovered because of his profile on iNaturalist, where he teaches me how to take photos of these cool little insects. I'd like to highlight 2 other kinds of organisms that I have come to know thanks to iNaturalist that are all over the place, but I just didn't have a chance to see. Anybody know what this critter is? Yay, it's a slime mold. So the sort of yellowish network is one organism, question mark, a colonial organism. It's kind of an interesting mystery. They can live as a single cell. They can come together as a group of cells. This is macro scale, right? So this is a couple of inches across. The little white pellets are food. They love to eat oats. So here's a time lapse of this kind of colony pseudo single organism going out and finding food and then centering in on it. Some of these slime molds are particularly famous that if you put the food in strategic places, the slime mold will go out and find the food and then make a network that's connecting the food most efficiently. So if you put the pieces of food where the major urban population centers are in Tokyo, in about 24 hours, your slime mold will have solved the Tokyo rail network. And if you overlay this image from the slime mold on top of the Tokyo rail network, it's almost identical. There are some mountains in the way that the slime mold didn't know about, but more or less, this is some of the most efficient connections for this organism. Slime molds are in your backyard, if you've got a backyard. And if you haven't got a backyard, we've got lots of parks where they're at, right? Slime molds can be remarkably beautiful. What I just showed you was their sort of creeping around form, right? But when they are getting ready to reproduce, like mushrooms, they reproduce with spores that are very light that move throughout the air. And those spore-bearing structures can be remarkably beautiful. Some of them seem to defy gravity in the way they suspend upward. And all of these photos were taken by Alison Pollock, who's a retired mathematician. She did the math and she's like, you know what? I'm just going to take photos of slimy things in the woods. I have never met Alison, but I adore waiting for the next photo that she is about to send up. All right. She has a quote on her iNaturalist profile that says, "A wet forest is my happy place," with a smiley. And she has her modified camera. She actually has one that has a microscope objective on it to get these really up-close images. And so Alison offers great insight into these organisms and to identifying them in California especially. But there's another person who does a lot of slime mold photography and identification. So here are more of her, this is the next person's photos, right? Really cool, colorful structures. This is Sarah Lloyd, who was basically a homesteader in Tasmania. She liked bird watching, kind of like me with the reptiles, right? And then she's like, I started noticing these slime molds, and then I started documenting them. And she has now realized that Tasmania is one of the best study places for slime molds because of her work on her farm there. She's written a book that has at least 4 editions where the slime mold creeps, and has some wonderful photography as well. Super available on iNaturalist. Another person I have not met, but who has influenced me greatly, is Luann Roberts. Now, Luann Roberts is on a dock. And Luann Roberts is on a dock with her excellent hair and tattoos, including the tattoo on her finger, which is a scale measure bar that she uses when she photographs images of sea slugs or nudibranchs. And Luann is prolific at this. And now that I was moving to the Pacific Northwest, North Carolina doesn't have that many sea slugs. We have some, but not like a ton. So when I saw a lot of Luann's photos, I was like, I'm in the Pacific Northwest now. I got to look for some of these sea slugs. But where do I look? When do I look? How do I look? Luann had blogs about it. She has this wonderful series of blogs that she writes, and she's very poetic. She's very lyrical in the way that she writes. She is not analyzing these organisms for number crunching. She's seeing the ecosystem sort of breathing and flowing from year to year. And I'm constantly reminded by her to sort of slow down and not seek the next Pokemon kind of species, but like just observe what is going on around you. And I really appreciate Luann's perspective for that. So here's an example of one of the most common sea slugs around here. I took this photo 2 days ago. I went to Point Defiance and I found like a dozen of these, the horned nudibranch. I also found a couple of these, which I'd only seen once before in a completely different place in Washington on the Olympic Peninsula. And I found like 6 of them, which I usually don't find at that spot. So I was really glad that I went a little adventuring for y'all. So through iNaturalist, I knew some good places to go and look for some of these critters. Here are some other species that I have seen in Pierce County of nudibranchs. The festive tritonid is probably one of the bigger ones. It's probably like adults like that big, but a lot of the other ones are quite small. Right? And in total, just in Pierce County, I've observed 22 species, which is not all of the ones that, for example, Luann has found, or some other people in this room, perhaps, but it's a good number. And I knew where to go and look because I just go on iNaturalist, and I'm like, where do I go for the sea slugs? And there's a ton of dots on the map at Point Defiance Park. So I just hang over the end of that dock and hope that I don't hit my head on a buoy or a boat and look for them there. Here is the blog that Luann runs, and I just wanted to put some of her voice in here. It's called Only Slugs. You can subscribe for free. Again, very lyrical. I really appreciate her observations. And one of the things she posted recently was, when I first started crawling around docks looking for sea slugs, and she's been doing it forever since, and so will I, right? I had absolutely no idea what I was doing. I didn't have a project. I didn't have a research plan. I definitely didn't have a data management story. But what I had was curiosity. And again, I want us to remind, like, you had the curiosity to come here. Thank you for spending some of your time with me on this. Whatever organism it is out there that brings curiosity to you, I hope that iNaturalist can be one of those tools to help you put a name on it, maybe find the unicorn horn that it's got on its face. Because that can build up to a fly guide, to becoming a museum curator, or in Luann's case, to having seen over 10,000 different nudibranchs in Washington state alone. So she has this data. Yeah. And she's not trained in this, but it became something she cared about. And she realized people weren't observing them the way that she was, and she could document it and generate data. So that's the last part of the story that I would like to share with you. We have sort of those traditional science ways of putting up a sheet or doing a beat sheet or using microscopes or whatever for gathering information on what species are where when they are there. But if you have everybody who's got a cell phone in their pocket start just taking a photo of whatever bug or insect or flower that they see that is interesting, that data set grows rapidly and we can learn a lot about it. There have been multiple species that were never documented by Western science discovered from iNaturalist photos. There was a teenager, I think in Peru or Ecuador recently, she took a photo of a butterfly and people were like, this is a new butterfly that has not been named by Western science. Right? There was a moth in New Zealand that had been recorded in New Zealand like 100 years ago. And so we had a pinned museum specimen, and I'm good. Fantastic. And then this tourist was like, "Oh, there's a little tiny bug. Let's take a photo, put it on iNaturalist." And everybody freaked out because they're like, "We haven't seen this insect in New Zealand for like 100 years." Right? You don't know what kind of contribution you might make, but the more of us that do, it's great. And sometimes even the really common organisms can be valuable as well. Right. So even if we're all taking photos of mallard ducks and Canada geese, we start to find patterns in those data. So here's an example. Researchers have looked at these two butterflies. On the bottom in the sort of the purple red color is data from about 1920s. Above is iNaturalist data more recently. And the horizontal axis is when during the year those butterflies were observed. So what I want you to see is that the iNaturalist data has a wider range of time throughout the year than those researchers back in the 1920s who were looking for the butterflies specifically. So now we know more about when and where these butterflies can live because more people have been observing them. We can, for example, use iNaturalist data to track when species arrive to a new place for the first time. So the spotted lanternfly is a gorgeous, pernicious insect that in North America we do not want. And you can see from this animation that I should be able to loop for you if I just keep clicking it, that it arrived in northeastern United States and over several years has spread out rapidly. All of these data were just iNaturalist observers being like, what is this cool bug I've never seen? I've been living here for 80 years. I've never seen that bug before. And they upload it, and now we know how this insect is spreading throughout the United States. So we can track where things are found and when they are found. This is another example of an invasive mosquito in Europe. It is an Aedes mosquito, so it does transmit diseases to humans. And so as global climate gets warmer, maybe these areas farther north are more hospitable to these more tropical mosquitoes. And so researchers can say, we have a much wider network to monitor for these disease-transmitting mosquitoes because people are putting their photos on iNaturalist. And they can also do things like look for when in the year the mosquitoes are most common. So if there's a peak at different times in different places, they can focus their intervention efforts then. There are over 7,000 research publications using iNaturalist data, right? And most of the people contributing, they're just like, high schoolers, retired mathematicians, right? People like you, some of you might already be using iNaturalist, but together we build a data set that is phenomenal and allows us to answer a lot of really cool questions, right? Things like species distribution and ranges, I talked about that, climate change, environmental impact, discovering new species, education and outreach, and so on and so forth, and so on and so forth. So I wanted to know, is that an unusual flowering time in Washington state for Salal? I could go to the iNaturalist data, I could filter by Washington state, and I could say, tell me, have people marked these photos as flowering or not? And iNaturalist shows me the graph on the right. And what I see is I forgot to give you the key, sorry. Red is flowering. So for only the Washington state observations, it seems like they usually flower April, May, June, July, and mine is kind of unusual. But what you might not see because of this projector is that there is a gray background. And those are all of the Washington state observations that nobody has annotated flowering or not yet. So even if you're like, you know what, I don't want to put my cell phone data on the internet, there are ways to obscure your location, by the way, for privacy. There's a version of this app that can be used without logging in at all. So if you have like young kids and you want to do that privacy thing, they don't have to log in and they can still get a suggested ID. There's lots of opportunity for people to sit at home and be like, oh, that's flowering. I can contribute to the data. And maybe we'll see, for example, if things are flowering earlier in the year as the global climate changes. I love iNaturalist. Since I have found it, it has genuinely improved the quality of my life, helped me do a lot of things, learn about organisms that I otherwise would not have, slow down and appreciate them. It has given me names. What is this thing? It has told me where I can go and when I can go to find things. And it has given me a rich number of connections with people, right? People who are just curious about the natural world around us. So when I was trying to think of what to do for one of these Grit City Think and Drinks, the only thing that came to mind was I want to share it with more people, right? I think that this is a phenomenal tool on par with something like Wikipedia in spreading knowledge for everybody. So I want to in particular let you know about an event that is coming up, a global event specifically for iNaturalists. It's called the City Nature Challenge. And the City Nature Challenge is, again, all throughout the world. And it's contributing to some of those data points, specifically focusing on urban areas. So like you don't have to go somewhere crazy to go find the ostrich or whatever. You can find the cool springtail or slime mold in your backyard here in Tacoma. We're having one for Seattle and Tacoma. The event is the 24th through the 27th. And any image that you upload that was taken during that time and goes to iNaturalist with the GPS coordinates in King County, Snohomish County, or Pierce County just automatically goes in. Like you don't have to enroll or any of that. If you are putting in observations, they will be counted as part of our project. But if you're like, I'm still not sure about this, we are specifically on that Saturday having an event on UW-Tacoma campus. So I will be here. Dr. Jeremy Davis, who is an entomologist, is going to be here. We're going to be looking for bugs and cool insects and stuff on campus with our little iNaturalist and taking photos. And I'm just excited to share that with you. So this is one of the ways that you can help participate with the City Nature Challenge. But again, you don't have to do anything official. Right? If you've got the account and it's during that time window in that area, it will automatically count for our data so we can learn more about the natural world around us. There are two other events if you are interested. One of them is that Tuesday, the 28th, we have a guest presentation from someone from the University of Central Florida, Dr. Sonia Stevens. And she's going to be talking about what I talked about, but not from the biologist point of view, from like the point of view of, she does like communications, right? So we're interdisciplinary here at UW-Tacoma. We love bringing all the different disciplines together. And she's recognizing that when people use iNaturalist, they start looking at the world differently. So she's giving a talk that's, now I'm always looking for that butterfly. If you are interested in hearing it from her perspective in sort of the communications of scientific information, how it changes our lives. And lastly, there was one more event that I wanted to announce. What was that other one? Oh, yes. Luann, I will finally get to meet her, at least online. She's giving a talk about how to do sea slugs, how to find them, how to respect the environment when you're doing that. And that one's going to be digitally remote also on 28 April. So that is what I have for you. I appreciate your time. The hands-on portion of this event is that you may have noticed they have a lot of specimens out in front of you. So what we're going to do is, as Jim said, there's like a quick break time now where I can go outside and let out my scream and then come back in looking all composed. And then if you want to try out iNaturalist, I can help you. Almost all of the organisms that I have brought that are large should be pretty identifiable almost immediately. But I did also bring some slime molds and some sea slug. No, I didn't bring sea slugs. They would not survive. And some springtails. If you would like to see them either through the microscope or just sort of hold them up in the Petri dish and have a look at them. Okay? Cool. Thank you, everybody. This iNaturalist is a social network, and its stated goal is to help people document their interactions with nature. There is an ability to follow people to see when they post. And if you know somebody's handle, like mine is Heinz, you can follow that person and see when they update. So it is very common. I have some friends from when I was in middle school volunteering at a science museum that we just have followed each other's trajectories. And he's in Florida and he keeps posting snakes, and I'll just like put a smiley face on a bunch of his snakes. And every once in a while we trade back and forth jokes about it. But interestingly, a couple of years ago, iNaturalist was actually voted, I think it was by the New York Times, as one of the friendliest places on the internet, because there isn't a lot of the back and forth. Like, it's a social network, but the whole thing is, look at this cool fungus or whatever I found. What is it? Right. And so the dialogue that happens back and forth is a lot like I was showing. It's a spider. Oh, here's exactly the species. Oh, cool. I didn't realize it had that face thing. Right. Or it's a spider. Oh, I could have told you the species if you had given me a photo of the belly instead of just the back. Great. Next time I'll try and take a photo of the belly as well. So most of the back and forth is like, how do I know it's this species and not that species? Oh, it's unusual to be here this time of year, that kind of thing. And there's a lot of sharing of resources and publications. As being a professor at the University of Washington means that I have access to our library, which has extensive connections with research publications. And if you don't have those connections, getting access to just one publication can be like $45, $50, $60. So I've I've had people be like, I think this tarantula you took a photo of in Ecuador is this new species that was just described, but I can't get access to the paper to verify that its technical description matches your photo. And I was like, guess what, buddy? I have access to that paper, and I've emailed it to people. I got our library to find a German PhD dissertation, and they sent it to me, and I scanned a bunch of photos and I sent it to somebody else for these little diatoms in water, and they were like, I've never been able to get a hold of this book. They were so excited to have 100 pages in German of like sketches of microscopic organisms. So it is very much a social network. You can follow and unfollow people. There's a blocking ability too if somebody gets really persnickety about it. But it is very focused on the sharing of nature. Yes, we'll say. So what happens if you spot an invasive species? There's a lot of story that can happen there. When you upload an image to iNaturalist, the software assumes that you got the date and the location correct. So there are some default locations where if you don't enter in GPS coordinates, it will plop it down in the west of Africa in the middle of the ocean. And so there are like hundreds of thousands of observations in the middle of the ocean there. And we just all know, oh, that's a person who didn't bother to put in coordinates. So what often happens is the first thing people will be like, is this the correct location? Right? Because this species doesn't occur here. And people will be like, oh no, that's where I saw it. And then we'll be like, okay, this could be a problem. And so the next question, I'm going to show you an example. Okay. So here's an assignment I gave my students in biogeography last week. Anolis are a species of tropical lizards. They have really cool grippy pads, but they're not geckos. Here's the map of the world. Here's everybody on iNaturalist who put an image of it. They're native to Central and South America with one species that is native to North America. But you can see that there's actually quite a big range in North America. So if I go to New York State, I will see that two species of anole have been observed there. The green anole on the right, which is native to North America, but should not be able to survive in New York City's cold winters. This is like a tropical lizard. And on the left, the brown anole, which is not native to the United States. And so people are like, anytime they see them in New York City, they're like, are you sure you saw this lizard here? Very often, the lizard was spotted there, and that was verified. If somebody was like, oh, no, I saw this in Florida, you can go in and you can check the data and be like, the location is wrong. And then it's still visible on iNaturalist, but it's not in the scientific data set. So there is a way to curate the data. These are all the ones where people are like, no, I saw it for real there. And look what you can see in the photos. This one, that one, that one, that one, that one, this one. Almost all of these photos were taken at garden centers where they had imported ornamental shrubs from like Florida, Texas, Central America. Right. So we can monitor the introduction of new species through things like this. And when it's really unusual, people will jump on it and be like, this is an unusual record. There have been instances of iNaturalists being the first to find a true invasive, about to get established species. In those cases, happily, this is a tool that, for example, the Department of Ecology in Washington State monitors, right? There's a list of species where they're like, if this shows up on iNat, we need to look for it. For example, murder hornets, right? When those were the thing in 2020, if you saw a hornet, a lot of people were like, was it a murder hornet? I don't know. So you just take a photo of it, you put it on iNaturalist, and everyone tells you on iNaturalist, that's not a murder hornet. You're good. But if people did see a quote unquote murder hornet, Washington state government was looking for that and would know and be much more alert because there are more people with cell phones than there are people working for the Department of Ecology. So yeah, if you find something and it is verified in a time and place that it is not expected to be, then we can get that information to authorities. But also we can just have the helpful identification in the first place. Okay. And then yes. Amazing question. So I had cited that over 7,200, 300 different papers have used iNaturalist data. Not all of those have used only iNaturalist data. In fact, not all of them have used even mostly iNaturalist data. But in iNaturalist, for every observation that you put up, you can hide it from other people. You can obscure where it's from other people. You can hide the date. Like, you have some control over that. You also have control over the copyright for the image. So you can say, I'm just releasing it into the wild, or you can say, this is strictly my copyright. In the event that you allow flexible copyright, the record is exported over to the Global Biodiversity Information. What's the F in GBIF? Foundation. Fantastic. Thank you. And this is a much better, it's a subset of the data that's more thoroughly vetted, right? So we've weeded out the cats and dogs that are domesticated. We've weeded out the things that are in the wrong place, that sort of thing. It's a more vetted set. And that's usually the data set that's going into the research. So you only go into GBIF if you said, I'm willing to share my observations over there. And every single record is tied back to an iNaturalist account. So every single observation, you can cite that person with whatever name they have put on iNaturalist, whether it's, I love kittens or it's Heather Morgan Heinz, whatever. And that can be cited. So you will see people in these research publications say that this person found, and they are sharing the image with us. The flip side of that is I have been skirting around the issue of how it suggests what the organism is by looking at your photos, right? This is computer vision. It's a kind of AI. It's not a large language model. It's not generative AI. Rather, it's a classifier. But it is one of the best data sets for visual classification that's accessible out there, right? There's 300 million observations. Many of those have been checked by at least 2 different people to tell it down to species. So the software is offering you a suggestion based on those people's previous observations, but the data sets are also being used to train better and better computer vision. So that is, I think it is quite a cool use of the AI, and they have done calculations on how much ecological impact it has, and it's running on basically 2 computers in their office in San Francisco. So it's not a huge intensive thing the way generative AI is, but contributing images to it does add to that large data set. And so there is attribution where we can to each of the individual data pieces. But if I'm doing a data set of like 10,000, it's just like, thanks to the 10,000 people on INAT who uploaded dandelion photos, I used your data. Yeah. Yes. Oh, yeah. Sure. So we have a social scientist here who is asking about the process of truth seeking. And one of the things I like about iNaturalist is that it is somewhat self-correcting. So you will have the occasional bad actor who uploads, they go to Google and they get a picture of chimpanzee and they upload it and they identify it as their brother or whatever, or the other way around. They upload a photo of their brother and they say it's a chimpanzee, right? That happens. But everybody else is using iNaturalist will notice very quickly. They'll be like, that is not what that is. And there are data qualifications. So on any of these records. No, come back. In addition to the location, things like, is it adult, dead, alive, that kind of thing. There is data quality here. And this is where people were like, is it wild? Is it not? They're kind of going back and forth. And the record's never done, right? Anybody can go up and everybody has one vote, right? That includes Rod, the curator at the museum, in the Burke Museum, right? And it includes just somebody who just wants to click whatever they want. Right. So if Rod says it's this spider species and somebody else just makes up something, they will be held equally. But the longer records are on iNaturalist, they tend to converge on accuracy remarkably well. For birds, it's like you put a photo, even like a halfway decent of a photo of a bird up there, you're going to know what species it is within like 30 minutes. A lot of other organisms are a lot harder, fungi, for example. But it is, because it's egalitarian and everybody sort of gets that one vote, they do converge. And for many groups, it's like better than 90% accuracy, which is remarkable. And when people disagree, again, they're pretty collegial about it. So some people will be like, I know it's not that because of this. And then you can go and you can withdraw your ID. I often do identification at a very high level. So I type in very quickly. I'm like, if it's a bird, I just put aves because that's the big group, right? And if it's a bony fish, I put Actinopterygii, and those both start with A. So I have multiple times identified a fish as a bird or a bird as a fish because I just hit A and then enter really quickly. And people will be like, what are you talking about? Like, oh, and I need to go back and fix it. So it is there, and there's also a recognition of the lower limit of identifiability. So many times you have not taken enough photos of that spider to know what it is to species. And so people will say you can't go that specific, keep it at genus level or family level. And that is understood to be the taxonomic organization of these data. And I would say that while there's griping and go back and forth about it, the goal there is we want to be accurate with the observations. Yeah. Yeah, it's definitely a process. And I think you will hear more about that in the talk that's on the 28th too. Yeah. There wasn't out there. Hi. Someone like there is a problem of reinforcing incorrect. For example, earthworms it is not the common earthworm. The AI only knows about the common earthworm. There are hundreds of species of earthworms. It's going to tell you it's the common earthworm. It's not the common earthworm. There are other species. So there are taxa for which it is overconfident, and people are reinforcing that. But there are people who are being super diligent going back and fixing it. One example I didn't give you with the springtails is we had the wrong name for one of the globe guys for a long time. And then a researcher who I haven't met in person, but is a friend of mine now through iNaturalist, he was doing microscopy work up in British Columbia, and he's like, this is the wrong name. And he has a flock online of like a dozen high school students who really love springtails. And they're like, all right, now that you've told us what the new character is, and they went through thousands of observations and they fixed them all. So it is a self-correcting system. The other thing I wanted to show, what was I going to show you? Oh, how do they know the data set is good? They run an experiment where so once an observation has two different people agreeing at species level, it's said to be research grade, and then it's removed out of the 2ID pile, but it's still accessible. So what iNaturalist does every couple of months is they take from there, and then they get people who are experts in the group to go back and look at the research grade ones and say, do you agree with the research grade observation? And that's where my 90% for some taxa comes from. So yeah, it's those verifications on how accurate it is against experts, and they're not always right. And I think dandelions and earthworms are real bad ones, but yeah, like birds and stuff is pretty much perfect. So iNaturalist is for observations of all living things or evidence of them, and that includes microscopic organisms. That includes things like footprints, song calls, and so on. The accuracy of it is a combination of how many records have been identified on iNaturalist. So more records means more data to draw from. How easily those records are identified to species from photographs. So for example, bacteria, you can't look at them and tell them apart. These guys have taken microbiology. They're like, yeah, right. We do it with like the kind of sugar that the bacteria can eat or not. And so that evidence is not really readily in here. And so that's another example of a taxon where identifying a species is unlikely. But having a lot of records, and if you have the useful evidence of it on iNaturalist, even microscopic stuff definitely gets identified. The diatom example that I gave where I scanned the book and sent it, there's a couple of people who really like diatoms, and they ID them for sure. Yep. Or larval stages of like sea urchins and crabs and things. There are people out there who have that expertise. Yeah. I was about to say something, and then I realized my expertise is suddenly becoming slime molds and sea slugs. So, hi, Alexander. Mm-hmm. Sure. Mm-hmm. So Alexander's asking about how do these data contribute to our understanding of evolution, in terms of like discovering new species, finding places where they are or they aren't. And are you also asking about like fake generated data going in, or? Yes. Sure. Yeah. So we don't have time machines and we can't go back and look at what our ancestors look like for sure. But if we look at a lot of our close relatives, we can make a good guess, right? If you give me a bunch of cousins that are blood related, I'm like, I'm pretty sure I know what your grandmother's eye color was just from like looking at this many cousins, right? And we can go farther and farther back in time, less and less specific the farther we go back in ancestry. But as we find more species and understand their behavior, where they are found, what they look like, we can make more inferences about who they are related to. And that tells us about what branches of the tree of life are really diverse or less diverse. And then we can start asking other questions like, why is there only like one kind of platypus, but there's all these kinds of beetles? Right? And we would only know that if we identified a bunch of the beetles. So some of this is just the basic identification of species, where they are, and then we can try and make guesses on how they related and their ancestry once we have some of those basic data. Sure. So thank you for pointing that out. What I was talking about there was the big branches of evolution where we have new kinds arise. Like this group split into I don't know, the birds versus the dinosaurs, what have you, right? But there is also the sort of small population level where everybody who is in one species isn't the same as each other, right? So if we have a whole bunch of moths and some of them are a little bit darker and some of them are a little bit lighter, who's going to survive in this environment, right? If the darker moths are surviving better, then they make more babies, which means the next generation, there's just more of them that are dark than light, or vice versa. And so by having many, many more records of the variation in populations, just by virtue of having more photos, we can start to notice shifts in population that we might not have seen before. And one example of that, I believe the moth example I gave you was not arbitrary. It's one of the more famous ones. The Industrial Revolution in England put soot into the air, and then the birch trees that have white bark started having sooty dark bark. And so the moths that were light colored and used to blend in now stood out and they got eaten away, all within one species. So that species, more and more of them proportionally in the next generation, the offspring were darker, right? As the air got cleaned up, we could see a shift back towards some of the lighter as the barks of the trees were not covered in soot quite so much. That same species of moth has been documented a lot on iNaturalist, and we can continue to look at what proportion of the population is which color or other color. And we can only do that by having a really large number of observations. And that's the power of everybody with the phone in their pocket doing it. Right? What we used to do with that before something like this is museum collections where you'd go and you would just kill 200 butterflies and pin them and then store them in a museum and hope that somebody eventually goes and measures all of their wing colors. Now we can document that with photographs in a way that doesn't harm the animal and gives us a much larger data set. So it's incredibly valuable for that if the images document the thing of interest. Also interactions. So like what insects pollinate what flower? With the spotted lanternfly I showed you, one of the major things that we are looking at, and other researchers are looking at in iNaturalist data, is what native organisms are starting to eat the spotted lanternfly. So if somebody has a photo of a blue jay eating a spotted lanternfly on iNaturalist, that's really important for us to know. So we also look at interactions, not just within one population. I really wanted to thank you for downloading that. But I did want to give you a chance to try it, right? If you've got it, just take photos of the things that I've got here and you can see how quickly it'll tell you what those organisms are. And if you want to see some of the slime molds and stuff, I did bring some for you. Yes. Yes. So Seek is another app created by the same group. It is trained on the same data set. Seek is a little bit more user friendly. Seek, you do not need you do not need to create an account, and it will give you a suggested ID. You can get little badges for yourself, like you saw 10 kinds of insects, without ever having to log in or upload it anywhere. Right? So even though it is using the computer vision, that algorithm is in the app. It is not sending it to the internet. The internet's telling you what it is, and then it sends that back. It's all done in the program. So I would recommend Seek if, for example, you got a young kid who wants to use this, and you're like, you know what? I don't want you maybe accidentally putting your GPS coordinates in here. We're just going to give you Seek. Right? Then you have all of your record of the cool things you saw. It identifies it for you, but there's a layer between you and uploading it. If you want, you can put your Seek observations into the iNaturalist as well. That is possible. But yeah, the push is more for iNaturalist for the power user, I suppose.